I have the great pleasure now of being joined by Highlight Symposium speaker, Dr. Richard Carson. Thank you for being here.
Happy to be here, as always.
So I want to talk about something that I think is wonderful and groundbreaking and so necessary, and that is neurology research as it relates to nuclear medicine.
The beautiful part about what we can do in nuclear medicine is to be able to combine targeted radiopharmaceuticals that are going to look specifically at proteins or pharmacology in the brain that is specific to a given disease, and then build that with exquisite instrumentation that has just continued to just blossom in terms of the quality of the images that we see, and therefore the diagnostic quality that we can get from those kind of images.
So let's look forward and say, how does that translate into clinical practice? Because that's where it's so meaningful.
Right, and so we're seeing that already emerging in the area of dementia, Alzheimer's particular, and even in the more other neurological disorders related to that, to be able to have the tracers that bind specifically to these misfolded proteins. I mean, the classics in Alzheimer's are amyloid and tau, and we've evolved some very, very nice tracers. It's a long process. I mean, the idea is of even early on, the first measurement tool for amyloid, carbon-11 PIB, over 20 years ago. I mean, people are still working on improving, fine-tuning those tools. But now, as treatments become available for Alzheimer's disease, the ability to identify patients as early as possible really requires exquisite radiopharmaceuticals with great sensitivity, the ability to identify these issues in the brain in very small regions. And then when we combine that with elegant new instrumentation, we have incredible tools that really save the lives of patients.
And educating people seems to be the key in that, because you really do have to get to patients super early. So people with a family history?
Right, exactly. And sometimes it's family history there, but even then there's so much of these disorders are not understood about what the etiology would be. But I think the area that's going to show a lot of excitement very soon is in Parkinson's disease. There's incredible effort, and a lot of that was support from the Michael J. Fox Foundation, to be able to develop a tracer for the misfolded protein in Parkinson's alpha-synuclein. And that's going to give that jump start for the clinical trials to be able to identify those patients who are most likely to benefit by treatments that will be affecting those early loss, in this case of dopaminergic neurons.
So when you talk about building on decades of research, and when we think about nuclear neurology, what do you see us really honing in on in the next, say, 5 to 10 years?
Right. So I mean, what we've been doing for many, many years still works. And I think the idea is that as we learn more about what the right molecular targets need to be, and then how to turn those into successful radiopharmaceuticals. But it's still challenging. I mean, there's a lot of elegant work in the molecular modeling to understand the docking of these molecules into these proteins, but that's only part of the story. It depends a lot on how these tracers get taken up throughout the body, how they get metabolized. How does that work to produce the contrasts as well as the quantification we need to be able to make an accurate measurement of what's going on in individual patients. Long way to there's always room for improvement, but we have, once we get that starting point of here's something that seems to be working, then the field really jumps in and we really move that into something that can get used in clinical practice.
You are the recipient, 2024 Henry Wagner Image of the Year.
Thank you very much. We're very excited to receive it.
So tell us about that image and what it means.
So we have been fortunate to build a collaboration over the last now 5 years with our team at Yale University, with the team at University of California, Davis, that pioneered the total body systems, and with our industry partner, United Imaging Healthcare. And we were able to build a true next generation brain PET system. Now, brain PET systems, the last best machine was developed over 20 years ago, the HRT, High Resolution Research Tomograph. A very good machine, but there hadn't been the impetus to be able to push the technology forward, to be able to truly image extremely small structures within the brain. Now, lately, over the last 10 years, there's been a lot of interest in the society and related societies about developing this technology. But here we were able to put together the experience we had in brain PET imaging with the HRT, the experience at UC Davis with doing high sensitivity imaging, and the experience of United Imaging of actually building a production system. And we were incredibly excited to get funded by the NIH Brain Initiative to be able to support that work, and then be able to develop and deliver the system pretty much on time, which is pretty much unheard of. And now it's very exciting this year. What we've been able to do is use the great array of radiopharmaceuticals that we've been having to study the brain, but doing that at ultra high resolution and ultra high sensitivity.
So talk about how that's going to revolutionize patient care.
So what's incredible there is if we can truly see that branch out beyond the research labs, but actually into the clinical labs, it's going to let us being able to make some of these exquisite measurements. So one area would be in early Alzheimer's disease, where the deposition of the tau proteins in a very small region of the brain, the entorhinal cortex, to be able to measure that extremely well, to be able to say, yes, this is a patient that would be appropriate for some of these new therapies that are coming out. Or even better, there's a lot of hypotheses about an even smaller region in the brain, the locus ceruleus. Can we start imaging that and reliably quantifying and say, yes, this subject is showing deposition of these proteins. Same in Parkinson's disease, looking in the substantia nigra and being able to measure these very, very small regions. But one other little subtlety that may come out of this, our machine is sensitive because like the Total Body Explorer, it's long, it's 50 centimeters, goes down to the shoulders, which means not only can we image the brain, we can image the head and neck. And I'm excited about our early data showing the improvement in cancer imaging in head and neck just by having ultra high resolution. It will let us see and quantify tumors, lymph node infiltration at a much higher resolution. And so far, I'm expecting that our oncologists are going to get very excited as they see the improvement in those images and the ability to affect patient selection and treatment.
So you're hinting at the future. What is your ultimate goal?
Right, so we have lots of great questions about what we can do in imaging the brain relevant to applications. So another aspect of the high sensitivity means we can inject a lot less radiation. And that opens up the ability to begin to do scanning in adolescents. So we have collaborators doing an autistic spectrum disorder, in schizophrenia, diseases believed to be happening in the teenage years. And can we begin to do those scans with ultra low radiation dose? Say the amount of radiation you get by, flying to Disney World and back with your kids. And that would be the place where now our tools will be broadening much, much more to be very, very relevant in diseases where it's not clinically needed at that point, but it could drive the research and then the tools to be able to identify appropriate treatments. And that is incredibly exciting.
It is fascinating. I see why you are the award recipient. Congratulations again.
Thank you so much.
Thanks
It's
for
a
being
great
here.
honor.